Namaste and welcome back to JEE Chemistry with Typical Monk. This is class 34, Biomolecules and Polymers. Carbohydrates, proteins, and nucleic acids are all built the same quiet way. Small units joined into long chains, and each link forged by a condensation that lets a molecule of water slip away. Two amino acids meet and form a peptide bond. Two sugars meet and form a glycosidic bond, and a whole protein or a strand of starch is simply that step repeated. The very same idea carried into the laboratory gives the polymers of everyday life. Polythene built by addition, nylon and terylene built by condensation. Learn the bonds, the reducing sugars, and the two kinds of polymerization. And this chapter becomes a set of keen, scoring facts. Today, biomolecules and polymers, one idea at a time. Here is what you will master today. First, carbohydrates and the glycosidic bond. Second, proteins and the peptide bond. Third, nucleic acids, vitamins, and enzymes. Fourth, polymers and their classification. And fifth, addition versus condensation. One idea at a time. Let us set the foundation. Biomolecules, the molecules of life, carbohydrates, proteins, nucleic acids, and lipids. Monomers to polymers. Small units join into long chains. The bond, a condensation, losing water, links them together. Master line, life and plastics are both built from repeating units. Now look at the picture, the peptide bond. The four classes, carbohydrates, proteins, nucleic acids, and lipids. The monomers, sugars, amino acids, and nucleotides joined into polymers. The peptide bond, two amino acids condense, losing water, into a CO and H link. The glycosidic bond, sugars join the very same way, also losing water, in carbohydrates. Now, the carbohydrates, the sugars, monosaccharides. The simplest, glucose and fructose, they cannot be hydrolyzed any further. Disaccharides, two units, sucrose, maltose, and lactose, joined by a glycosidic bond. Polysaccharides, many units, starch, cellulose, and glycogen. Reducing sugars, glucose and maltose, reduce tolerance and feeling, sucrose does not. Now proteins and nucleic acids. Amino acids, the building blocks, each with an amino group and a carboxyl group. The peptide bond, amino acids join, losing water, into a polypeptide. Structure, primary, secondary, tertiary, and quaternary levels. Denaturation, heat or acid destroys the shape, not the primary structure. Nucleic acids, DNA and RNA, chains of nucleotides carrying the genetic code. Now, look at the picture. The two ways to build a polymer. Addition, monomers with a double bond add together, with no byproduct. Condensation, monomers join by losing a small molecule, like water. Note the examples, polythene by addition, nylon and terylene by condensation. Natural versus synthetic. Starch, proteins, and rubber are natural. The plastics are synthetic. Now the polymers in full. A polymer, a large molecule of many repeating monomer units. Addition, monomers with a double bond add polythene, PVC, and Teflon. Condensation, monomers join, losing a small molecule, nylon, terylene, and bakelite. Natural and synthetic. Rubber and proteins are natural. The plastics are synthetic. Copolymers, made from two or more different monomers, like Buna S. Now vitamins, enzymes, and hormones. In vitamins, fat-soluble, A, D, and K, and water-soluble, B and C. Enzymes, biological catalysts, mostly proteins, and highly specific. Hormones, chemical messengers that regulate the body. Now match each monomer to its bond. Amino acids go to the peptide bond. Sugars go to the glycosidic bond. Ethene goes to addition polymerization. And nylon goes to condensation polymerization. 4 pairs, each worth memorizing. Now, how many of these statements are correct? Statement 1. Amino acids joined by a peptide bond. Correct. Statement 2. Sucrose is a reducing sugar. Wrong. It is non-reducing. Statement 3. Polythene is an addition polymer. Correct. So the answer 2 are correct, statements 1 and 3. The trap is sucrose in statement 2. Now, the two mistakes that quietly cost marks. The truth: addition polymers have no byproduct. Condensation polymers lose a small molecule. The trap: thinking nylon is an addition polymer. It is condensation. The second truth: glucose and maltose are reducing sugars, sucrose is not. The second trap: forgetting that denaturation destroys the shape, not the primary structure. Now test yourself. Answer in your head before the reveal. That is how G-speed is built. Here we go. Question 1. The bond between amino acids. The peptide bond. Question 2. The bond between sugars. The glycosidic bond. Question 3. Polythene. Addition or condensation? Addition. Question 4. Nylon. Addition or condensation? Condensation. And question 5. Is sucrose a reducing sugar? No. Let us gather it all in one breath. Biomolecules, carbohydrates, proteins, nucleic acids and lipids. The peptide bond in proteins. The glycosidic bond in carbohydrates. Reducing sugars, glucose and maltose, not sucrose. Addition polymers with no byproduct versus condensation, which lose a molecule. Natural versus synthetic polymers. Rubber and proteins against the plastics. Next class, chemistry in everyday life. Built from repeating units. Everything in this chapter is one idea seen twice. In the living cell, amino acids condense into proteins through the peptide bond, and sugars condense into starch and cellulose through the glycosidic bond, each link made by letting a small molecule of water leave. In the laboratory, the same joining gives the polymers of daily life. Ethene adds to itself with no byproduct to make polythene, while a diamine and a diacid condense, shedding water, to make nylon. Remember which sugars reduce. Remember that denaturation spares the primary structure. And remember addition against condensation. And the whole topic settles into a handful of certain marks. Next class, chemistry in everyday life. Sarah, David, Lisa, and John all began exactly where you are sitting now. One idea at a time, you will crack G. I will see you in the next class.